Okay, so let's get started then. The title of this webinar is Textile Wastewater Treatment Using Microbial Fuel Cells. I've been working with wastewater treatment, specifically textile wastewater treatment, for quite some time. And we are grateful for the British Council to fund the METRIS project. So what I'm going to do today is to share with you a few findings from our lab as they relate to the use of microbial fuel cells using the use of microbial fuel cells to treat textile wastewater. Now, some of you, I sent you a few questions at the beginning to gauge your understanding of this area, and from what you sent me, many of you are quite familiar with the area. So, but I also know that there are a few who have just joined now, so I'm not really sure. So I may cover a few basics, so I'm sorry if I cover a few basics which you're already familiar with. So the starting point is, what sort of contaminants do we have in textile wastewater? So the textile industry is one of those industries which uses a large amount of water in the processing schemes that they have. So it is estimated that for every kilogram of finished product, the industry uses 100 liters of water. So that usually ends up as wastewater, obviously. So what you have here are examples of operations involved in textile processing and the type of contaminants that are involved. So in sizing, you have starches, waxes added. Scoring operations, you have sodium hydroxide. When it comes to bleaching the fabric, they use hydrogen peroxide. They also add other things like organic stabilizers, sodium silicate. When it comes to masterization, they use sodium hydroxide. When it comes to dyeing, obviously they use dyes which impart color, but they also add loads of salt to enhance the attachment of the color to the fabric. And actually it's been estimated that for every kilogram of finished product, 0.5 kilograms of salt is needed. So you can see that there are quite a number of chemicals that are involved in textile wastewater processing, and these chemicals ultimately end up in wastewater, and so the wastewater needs to be treated. Okay, not just to remove color, but also ideally to remove organic compounds involved in this water, any suspended solids. Ideally, because salt is heavily involved in this industry, ideally you ought to be reusing the salt. Okay. So the challenge is that we cannot start releasing this wastewater into the environment because of the negative effects it could have. So it's been said that some of the dyes used are carcinogenic. The color itself, if we release it into the environment, is not aesthetically pleasing to look at, and also the color can affect photosynthesis in plants that live in water. So yeah, so there is a good reason for why we need to treat this wastewater. Plus, obviously, in water-stressed environments, it's necessary to recover some of the ingredients in this wastewater. So if you can recover water, that's fine. If you can recover salt, that would be fine. And also, if you can recover energy as electricity, as we are going to see, that would also be useful. Plus, obviously, the other driver for why we need to treat this wastewater are environmental regulations. So in many countries, the regulations for treating wastewater in general are very strict, and actually in some countries companies are forced to close because they are not able to treat their water up to discharge standards. So the need to meet discharge standards is another important driver. So what you see here is a table that shows the typical characteristics of wastewater from the textile industry. So you can see the pH is shown here, 7+12, but the pH depends on the type of dye that you are using in the, or dyes that are being used in processing operations. You have organic matter, quantified as biological organic oxygen demand or chemical oxygen demand. Total suspended solids, you can see chloride ions, lots of it, and obviously color, as well as some nutrients like nitrogen and even phosphate as well. So when it comes to the textile industry, azo dyes are the most common type of dyes used there. So the challenge is that these dyes go through activated sludge systems unchanged. And while there are physicochemical processes like membrane filtration systems, they produce a lot of sludge, and also they're quite expensive. So if you think about the usual wastewater treatment methods where you are aiming to remove organic matter, remove suspended solids, remove pathogens if they are there, recover useful resources, you can see that whatever, and if you have to do it as cheaply as possible, you can see that this type of waste stream is very challenging, especially if you are going to do it using microorganisms. The challenge you will have, first of all, is high salinities here. pH, high pH values are going to not be useful, suitable for certain microorganisms. And also, even if we have organic matter in here, most of this is actually recalcitrant, i.e. difficult to degrade biologically. So this is a very, very challenging type of waste stream to deal with. So this next slide shows you a wish list. So I've created this wish list. So this is the type of treatment system I would want to have for treating this type of wastewater. So I want a system that is environmentally friendly. So that means it doesn't use many chemicals. It doesn't use much electricity. It doesn't produce sludge or much sludge. I would want a system that is cost effective, because at the end of the day, it's economics that overrides many things. We want a system that is carbon dioxide neutral. We are living in an era where increasingly resources are scarce, so we want to recover resources from what appear to be waste. So can we recover salt? Can we recover water for reuse? Can we recover energy as electricity? You want a system with very low or minimum maintenance. A system that can operate at room temperature. You want a system that is fast. Okay? You want a system that can lend itself to continuous operation. Ideally with short hydraulic retention times or high organic loading rates. Your system should be robust to things like changes in high salinities, changes in pH. So your dye, your textile industry may be using acidic dye today, basic dye tomorrow. You want a system that is robust to changes like that. You want a system, obviously, that can achieve discharge standards, otherwise your industry would be closed down. But you also, when you're developing your system, you want one which you can easily scale up. So if there is a change in production levels, or if it's an industrial site and there are many other industries involved, can your system then cope up with increasing demand in terms of volumes? Okay. So obviously this is a long list of demands, and it's difficult to imagine a treatment process that can meet up all of this. However, what I'm going to show you in this webinar is that microbial fuel cells actually tick many of the boxes in here. So at the moment, there are quite a range of processes used for treating wastewater. So there are physicochemical methods like adsorption, but the challenge with such methods is that additional treatment is required, for example, to regenerate the adsorbent, and that also produces a sludge which needs to be disposed of. Then we have electrochemical treatment. Now, electrochemical treatment is, on paper, probably one of the quickest methods used to treat textile wastewater. I mean, if you read papers, some people claim they can treat, decolorize wastewater, textile wastewater within minutes. But it has some disadvantages. Obviously, you are supplying electricity, so that is going to incur a cost. And the other thing is that it doesn't remove organics. It doesn't remove organics at all. Then we have membrane filtration systems. So these ones also work well, but they are quite expensive to maintain. Chemical oxidation is another approach used. This produces, it's expensive and also quite difficult to control what the outcome is. Then we have biological systems. So biological systems in general are advantageous in the sense that they don't cost much in terms of money. In many cases, they're sustainable. But the problem with many of them is that the quality of the water that comes out of the treatment system is not good enough, at least compared to the other physicochemical approaches. Enzymatic systems, like systems that use lacase, for example, can work, and these are also being researched. But my argument here is going to be that we could use microbial fuel cell systems to treat, textile wastewater. So this next slide shows you a comparison of the cost of the different treatment systems versus the quality of the discharge standard. So you can see that many biological systems are low in price, but also when it comes to discharge quality, they are on this side of the diagram. So high quality discharge. To get high quality discharge, you need systems like membrane systems, but these come at a high price. So I think there is interest these days in developing new technologies, and this is where microbial fuel cells systems come in. I think my estimation at the moment, obviously the technology is not, well, there are a few demonstration sites, but in the main, the technology has not been commercialized. But my estimation at the moment is that microbial fuel cell systems lie here. So they are quite expensive compared to conventional biological systems like anaerobic digestion. And also they give a fairly good quality discharge of your waste stream. And especially if you can combine microbial fuel cells with enzymatic systems, then you could have a winner there. So let me talk about the working principle of microbial fuel cells. So many of you obviously are familiar with this, but just in case there are a few who are not familiar with how this system works, what it is, it's basically a biological battery So typically you have microorganisms, and these have the capability of exchanging electrons extracellularly. So usually these microorganisms are natural occurring, and they have the tendency of forming biofilms around surfaces. So if you had an electrode here represented as blue, these microorganisms can form biofilms and attach themselves onto it. So if you fed this microorganism with an organic compound, like compounds in textile wastewater, the microorganisms oxidize the substrate into carbon dioxide protons and electrons. And because there is no terminal electron acceptor in the anode, the electrode, because it's connected to another reaction here in the cathode, which is at a higher redox potential, the electrode acts as a temporary terminal, electron acceptor, not terminal, but temporary electron acceptor. So electrons are then channeled, as you can see in this animation, outside the external circuit to the cathode, where they recombine with protons and oxygen to form water. So oxygen here is acting as a terminal electron acceptor, and in many microbial fuel cell systems, the oxygen reduction reaction represented here is catalyzed by platinum. Although, one of the difficulties is that platinum is a noble metal and it's quite expensive. So, in theory then, you should be able to get electricity, provided you can increase the substrate change, the turnover rate of substrate here. And also if you can minimize the resistances to transfer of, for example, protons and electrons. So that's basically the working principle of microbial fuel cell systems. You can use other systems here to accept electrons and protons, so not just oxygen. So some people in many lab scale studies use ferricyanide, for example. The other thing which is important is to remember, looking at this scheme, is that if you were to scale this up, then it's going to become expensive to bubble in oxygen or air. And so one of the new developments in microbial fuel cell design is using what are the so-called single chamber microbial fuel systems, or air breathing microbial fuel cell systems, where this electrode is not immersed in a solution, but rather it's attached to this membrane here in the middle, and it's sort of air breathing. So what are the advantages of microbial fuel cell systems for treating textile wastewater? So the first one is that because you can potentially get electricity out, it can be an energy positive process. So effectively you could have net zero operational expenditure. Because it's an anaerobic based process, sludge handling costs, basically sludge yields are low, and hence there should be low sludge handling costs. You can scale it up in a modular way, and so this is useful because it enables gradual implementation and easy expansion. For example, if there's a change in demand, or if there's an expansion in your processing operations. It's quite a robust process, so it can withstand high salinities, high sulfate concentrations, and wide pH ranges. Okay, and if you compare, for example, to digestion, it's quite robust. It's also suitable even for small installations with low organic loads. So this is another plus, especially when you compare to another digestion. It can operate at low temperatures, and even if you have inhibitors in your waste stream, like for example high ammonium nitrogen inhibitors of anaerobic digestion, the system can operate quite well. So looking at just these advantages, you can see that microbial fuel cell systems have promise in treating wastewater for energy recovery in general, but also specifically for treating textile wastewater. So how do microbial fuel cells fit in normal treatment systems? I think it's not my claim in this webinar that you can have a standalone microbial fuel cell system for treating textile wastewater. I think the reality is that the system is likely to be integrated with other systems. As we are going to see, it has a few disadvantages. But I can see MFCs being placed after the pre-treatment step in a municipal wastewater treatment plant here. And that is because usually you have low COD or low BOD amounts there. On the other hand, if you had an industry, so if it was an industrial wastewater stream and it had a high COD, then microbial fuel cell systems are likely to be positioned after an digestion system, which helps to remove much of the COD. And then effluent from that can be fed into a conventional wastewater treatment system and ultimately released into the river or another water body. If the stream had low COD or had some components incompatible with anaerobic digestion, then that can be fed directly into a microbial fuel cell system. So these two scenarios indicate how I see MFCs being integrated with existing systems, existing biological systems. Now, when it comes to the textile industry, iso dyes are the most common type of dyes used. And these are characterized by this moiety here, N double bond N. So you have examples here like reactive black, acid orange, 7, direct red. So quite a number of dyes used in industry have this characteristic functionality or functional group in there. So what I'm going to do now is to share with you a few, by way of case studies, a few examples of studies we've done in our lab to basically determine the efficiency of microbial fuel cell systems when it comes to degrading iso dyes. So when you think about microbial fuel systems and wastewater treatment, the first question is, should you feed the dye in the anode chamber or the cathode chamber? You know, because if the dye had a high enough redox potential, in theory, it could act as a terminal acceptor in the cathode. And there have been examples in the literature where, for example, methyl orange has been used as a terminal acceptor in the cathode, in much the same way as people use ferricyanide. Alternatively, like in this example I'm going to give you, you could replace platinum, which is used as a catalyst for the oxygen reduction reaction, and replace it with lacase enzyme, because lacase can also catalyze that reaction. And also lacase has the property of breaking up these aromatic phenolic compounds, you know, like this acid iron 7. So, I mean, one can, you would expect that if you had lacase in the cathode, it can serve the dual purpose of not just oxidizing, catalyzing the oxygen reduction reaction, but also breaking up the dye that is in the cathode. Okay, so that is one approach. The other approach, which is one of the most commonly used approach, is feeding the dye in the anode chamber. That's where you have microorganisms. So the idea there is that the microorganisms degrade the dye via a mechanism which are not entirely known. I'll talk about that in a moment. Yeah, so there are two options. So this first case study, we carried out an experiment where we compared the removal rates of the dyes, the energy produced, how toxic the degradation products are when you feed the dye in the anode versus when you feed it in the cathode. I talked about degradation. I need to clarify what I actually mean by degradation, because, or biodegradation, whenever you talk about the term biodegradation, many people think about mineralization, i.e. the dye is completely converted into minerals like inorganic compounds like carbon dioxide, water, ammonia, phosphates, depending on what atoms are involved in the compounds, in the dye. So it can mean mineralization, but it can also mean other things. So biotransformation, where a part of the organic molecules is converted. So for example, you had chlorobenzene, and you knocked off the chloride substituent and became benzene. That's some kind of biotransformation. Okay, so biodegradation can also mean that, especially if the resulting compound is less toxic. The other meaning of biodegradation is fragmentation. So when you have a molecule like this one, acyl-7, and you break that bond, the N-double-N bond there, so you basically fragment that compound into two. So that can also mean biodegradation. Okay, so yeah, just to clarify, when I say biodegradation, I do not necessarily mean mineralization. So I was talking about, should you feed the dye in the anode or should you feed it in the cathode? So when it comes to feeding the dye in the anode, the mechanism by which this dye is degraded is not exactly known at the moment. You would expect that the dye is not taken up by the cells as a carbon source, mainly because it's big in size. So the expectation is probably that the dye gets degraded via co-metabolism. So you have a co-substrate, so the cells, the microorganisms feed on the co-substrate, and by so doing they may produce some enzymes which end up degrading the dye. So that is one suggestion. Okay. So what we have here is a mechanism where you might think that electron donors like formate or hydrogen donate electrons which are channeled across the periplasm, and then the dye on the outside of the cell gets reduced via some kind of enzymatic mechanism, in this case involving an azo reductase. So the reduction can be direct, or it can be mediated by certain kind of molecules which may be produced by the cell itself, or which may be added to the system. There is also another theory where it is thought that some non-specific reactions, for example involving sulfides, sulfides are commonly produced under dynamic conditions. So if you add sulfide, again that can interact with the dye, reducing it in the process. Okay. So we think that the dye in the anode is not actively taken up from metabolism, but it gets degraded somehow by a number of mechanisms which are not exactly understood at the moment. When it comes to the cathode, where you're using enzyme lactases for catalyzing the oxygen reduction reaction, you expect your substrate, which is the dye, to be oxidized with oxygen acting as a terminal electron acceptor. So that is direct oxidation. If the dye is so big and can't fit into the active site of the enzyme, then you need some kind of indirect oxidation where you have mediators like ABTS or other types of natural redox mediators. So natural redox mediators are better because they are less toxic than the artificial ones. Okay. So in theory, you could then add redox mediators to the cathode, and that could also enhance the oxidation of your substrate, of your dye. So for this first case study, the research question is enzymatic cathodic oxidation of azo dye is better than anodic reduction in microbial fuel cells, and we looked at COD reduction, color removal, amount of electricity produced, and also the nature and toxicity of degradation product to answer that question. So very briefly, we used acid orange 7 as a model dye, 100 milligrams per liter. We used H-type MFC configuration with working volume of 200 mils per chamber. We used Shewanella onidensis, MR1 as biocatalyst. The analyte was minimal salt medium, and we supplemented that with trace elements and vitamins, as well as a co-substrate as sodium pyruvate 1 gram per liter. On the cathode side, where we have the enzyme, we have acetate buffer, 100 millimolar, pH 4.5. The membrane was the CMI 7000 cation exchange membrane. We had carbon cloth electrodes, external resistance 2 kilo ohms. For the system we're using, we did some studies varying different external resistances, and we found that the value of 2000 ohms is where we get maximum power outputs. So that's why we chose to use that. Lacase was loaded in the cathode. It's freely suspended at a loading of 0.3 units per mil. The voltage was logged online, so we have a Piccolo data logging system. And I did various offline analysis, COD, GCMS to determine the nature of the degradation products, spectrophotometry to determine color removal, this being a pure dye, so you know when like acid orange 7 has a maximum absorbance around 484 nanometers, so you can use that as a measure of color removal. And also we did toxicity tests on the degradation products. So this test uses Vibrio fischeri bacteria. So these bacteria bioluminescent in nature, so if they are grown in the presence of something which is toxic, you expect to see a reduction in the levels of luminescence. We repeated the experiment 3 times, and then we did some statistical analysis. So this is the structure of acid orange 7. As you can see, you have a N double bond there. So regarding color removal, what we find is that when you feed the dye in the cathode, you get a faster rate of color removal, at least in the first 24 hours. And that sort of reduces, so the rate reduces subsequent to that. And in the end, after 4 days, there is no really significant difference in terms of efficiency of color removal. We think that the enzyme probably gets inhibited by the dye after the first day. And we've actually been working with various ways of trying to improve the stability and activity of enzymes within the cathode using, for example, various immobilization techniques. But what we also see, and this is something which is not quite commonly reported in the literature, is that for the solutions in the anode, that's the starting position of acid orange 7 in the anode chamber. It gets decolorized quite fairly rapidly within 24 hours. But if you leave that standing on a bench, say within another day, if we come back the following day, you would find that the color has sort of if you like, reappear. Not exactly the same, but since they look similar, you might think that the color has reappeared. Okay, so this is one disadvantage of feeding the dye in the anode. This kind of color reappearing or another similar color appearing does not occur when you feed the dye in the cathode. Okay, so based on this alone, then you might come to the conclusion, just looking at this graph, that feeding the dye in the cathode is then better. So we are interested to know what are these, what causes the color to come back. So this graph here shows you an absorption spectrum of different dyes. So asteroid 7 as well as Congo red initially and after decolorization and after recolorization. Okay, so the black line is the initial, the green line is the decolorized solution, and this color here is the auto-oxidation process. So you can see simply by looking at the spectrum that this line here, what I'm calling auto-oxidized products, is different. It's different from the initial. So it's not like the degradation products are recombining to give you the original compound, but it's something else. So similar trends with Congo red. So that's the starting position. Then this one, the purple one, is decolorized one, and then upon auto-oxidation, you get that. Okay, so we think that what is happening is some kind of auto-oxidation, and this actually we looked up some literature and we found a paper published some time ago, which indicated that aromatic amines, which are the ones you expect to get from if I may go back here. So if you break that bond, there, so you get aromatic amines. Okay, sulfonylic acid and 1-amino-2-naphthol. This 1-amino-2-naphthol especially has the property of undergoing auto-oxidation reactions, and that probably explains the reappearance of a similar color to what was in the initial there. When you look at COD reduction, we found that when you feed the dye in the anode, you get a slightly lower COD reduction compared to if you feed the dye in the cathode. And we also had a control where there was no dye in either chamber, and this gave a COD reduction of 78%. I have to clarify here, when I say COD reduction, the COD is not in respect of the dye alone. So we are looking at the whole microbial fuel cell system. So remember, when we add the dye in the anode, the dye is mixed up with a co-substrate, sodium pyruvate. Okay. When we add the dye in the cathode, there is acetate as a buffer, but also in the anode, you have growth medium there, pyruvate. So when I'm talking about COD reduction here, I'm looking at reduction on a whole MFC basis, the whole of it, including anode and cathode together. When it comes to electrochemical performance, what we find is that if the dye is fed in the cathode, you get a higher power output than when the dye is fed in the anode. But what is also interesting here is that the control run, which had no dye in either chamber, produced a higher power output. So what this tells you is that when you feed the dye in either chamber, the dye has some kind of inhibitory thing going on, whether to the cells or to the enzyme. When we did work out the coulombic efficiency and internal resistances, we didn't find big differences to explain the differences you see there. We also looked at the toxicity of the end product. So remember the Vibrio microtox assay, which uses bioluminescence of this organism, Vibrio fish array. So we took samples, diluted them, so concentration is increasing, and then we added them to the cells and then measured bioluminescence levels. So percent inhibition is percent relative to the control where there is no toxic compound added. So what we find is that the auto-oxidation products are really toxic. Far more toxic than those ones in the anode. And you see an increase in toxicity as the concentration increases. For the cathodic products, there's a slight increase, but not that much. So this next slide shows example what we think is happening in a chamber. So in the cathode, in the anode where you have microorganisms, we think that the azobone is broken up there, releasing these two compounds separately. And we've actually done HPLC studies where we have shown that these compounds appear. On the lacase side of things, it's a bit complicated on the lacase side because lacase acts in a non-specific way. But based on the GCMS data, we found these compounds were there, and the similarity was so high for these compounds, benzoic acid and hexanoic acid. And we pieced together a kind of mechanism which might explain how these compounds appear. So we think initially that bond breaks there, and then this next compound undergoes desulfonation, and then ring opening to produce, or ring cleavage to produce hexanoic acid. Similarly, you might get bond breaking there, giving you that, and then this can undergo ring cleavage, giving you benzene 1,2-dicarboxylic acid, which then can further be oxidized to benzoic acid. So this is just a putative pathway of what we think is happening on the cathode side. So take-home message from case study 1. So we think that that work suggests that when you feed the dye in the cathode chamber, it's a better approach than feeding the dye in the anode chamber. Many studies utilizing MFCs for textile wastewater treatment feed the dye in the anode chamber. So we get better CO2 removal effectiveness. We had faster color removal. There was no color coming back. There was greater power density than if you fed it in the anode, and less toxicity of the degradation products compared to the auto-oxidation products. So this next study involves a continuous flow. So I mentioned at the beginning that a good system should at least be operated continuously, especially if you consider the amount of the volumes of wastewater that are produced in the textile industry. And also, I think some of you may be wondering, oh, why did you use the pure culture? Okay, I mean, pure cultures have their position when, for example, you're trying to understand the mechanisms of what's going on, and you want to maintain the type of strain the same. But ultimately, I appreciate that you need to be using mixed cultures to treat these complex wastewaters. So this case study was run in a two-stage system. The first stage was a microbial fuel cell system, and the next one was an aerobic system. We used both a synthetic wastewater involving acid orange 7 and real wastewater, and this was obtained, so it's segregated waste obtained from the acid dye bath of wool. The COD of the real wastewater was around 1,000 milligrams per liter, but the composition was unknown. For acidorin 7, we use a concentration of 35 milligrams per liter. So this is just a schematic of what we had as a setup. So we had 3 microbial fuel cell systems, tubular in nature, and they were operated in parallel. So we feed in our dye, and the retention time in each of the microbial fuel cell system was 13.3 hours. The retention time in the aerobic system was 22.2 hours. We added 2 grams per liter of molasses as co-substrate to the dyes. When you work out the organic loading rates, they come to that 5.76 for acidorin 7, the synthetic one, and 7.11 grams of COD per liter per day for the real wastewater. The volumes for each MFC is around 400 mils, and this one is 2 liters. So we evaluated performance of the whole system based on color removal, COD reduction, and power produced. So this is just the same setup, but just to show you how it looks like. So these are the 3 tubular air-breathing microbial fuel cell systems, and this is the feed tank, and this one was the aerobic system. So with regard to color removal and COD removal and power production for the synthetic wastewater, this slide shows you that you can achieve very high efficiency. So 90% COD removal as well as over 90% color removal overall. What we also see is that the aerobic stage, represented here in green, does not contribute to color removal. However, the aerobic stage contributed to COD removal. So by combining both processes, in the case of this synthetic wastewater stream, we were able to achieve a good color removal, good COD removal, and a power density of about 48 milliwatts per meter squared at current density of about 200 milliamps per meter squared. When it comes to the real wastewater stream, we also see high efficiency in terms of color removal and high efficiency in terms of COD removal. In this case, the aerobic stage contributed somewhat to the removal of color. The aerobic stage also here contributed to the reduction in COD, and the power density was slightly higher, 54 compared to the synthetic wastewater, and the current density was much higher, around 550 compared to 200 milliamps per meter squared for the synthetic wastewater stream. So the last case study I want to talk about involves robustness. So this is another key issue when it comes to wastewater treatment. So is there a change in performance when you change the organic loading rate? How robust is the system if you operate it over prolonged periods? So these are important criteria whenever you're designing a system, obviously. So in this particular case, we used synthetic wastewater but in continuous flow mode. So similar system as before, but using synthetic wastewater. The volume in this case was 200 milliliters, and the volume of the aerobic stage was 1,000 milliliters. I have to go back here just a moment, just to tell you that we used, I didn't talk about this, the consortium used here to seed the MFCs was an adapted inoculum. So it was initially, we had a mixed community feeding on acidorin 7 for quite some time. So that's what we used to inoculate the MFCs. And the inoculum here for the activated sludge system was from activated sludge. So same principles here as well with regard to the inactive. So when it comes to color and CO2 removal, what we see is that when you change the organic loading rate, the performance of the system overall in terms of acid orange 7 concentrations, in terms of overall CO2 removal, is sort of maintained. So that's good, because when you increase the organic loading rate, you don't want the performance of your system to deteriorate. What we also see, we included an open circuit control, represented here in green line with the black dots. This here, you can see that the MFC stage and even the aerobic stage perform far better than the open circuit control. So I mean, some people sometimes argue that why don't you just use anaerobic digestion? Why use microbial systems when you can also recover energy from anaerobic digesters as methane? Okay, so you can see that pure anaerobic digestion fares worse than microbial system. So we think the decrease here in terms of the COD is due to removal of these compounds, the amines produced in the microbial system, as indicated here. So increase in loads, the MFC, we see a decrease in concentrations in the aerobic stage. So same sort of profiles for sulfonic acid. When it comes to power production, we've got about 52 milliwatts per meter squared of power at a current density of about 250 milliamps per meter squared. So general talk home messages then, microbial fuel cell systems have potential for textile, cost-effective and sustainable wastewater treatment. Okay, so the system has to be cost-effective. At the moment, they are unable on their own to achieve complete mineralization of dyes. There's also need to improve power output, but they are quite efficient when it comes to removing color. So there are a few outstanding research questions. So how best should MFCs be integrated with other treatment options? How can the cost of materials of construction be reduced? So, for example, platinum used as a catalyst. How can we develop better catalysts, cheaper catalysts? How can scaled up systems perform over varying conditions? So if you change the type of dye composition, if you're operating at different temperatures, different pHs, and how can the design of reactors be improved? So as part of metrics, for example, we have designed new reactors, single chamber systems like the one you see here, and we are trying to investigate how we could improve the design so as to further improve performance of the systems. So finally, I want to say thank you to a few people who have done the lab work. You know, they're the ones who produce the results. We have Priya here, who's going to do Harvard very soon. Yes, Fernanda, who is now a senior lecturer in Sri Lanka. We have Kyle Bowman and Mark, who are research assistants working on our matrix project, and my co-investigators, Ola and Taj Geshetas. Okay, thank you very much. So I now take a few questions. If we run out of time, I may have to respond to your questions via email.